Have you ever stopped to think about what's going on inside just one of your cells right now? It is a non-stop microscopic city, a universe in miniature that, for most of human history, was totally invisible. Well, today, we're taking a journey into that world. We're going to explore the incredible tools that scientists have built to peek inside, to see how human ingenuity literally made the invisible visible. This is a story of how we learn to see life's smallest secrets. Let's dive in. So right off the bat, here's the scale of the challenge we're up against. Your average human cell is maybe 1/100th of a millimeter across. A grain of sand is 1,000 times bigger. We're talking about a world so small it's almost impossible to wrap your head around. So the big question is, how on earth do you even begin to explore a place like that? And really, that's the whole game. Every single tool we're about to talk about, every clever trick and piece of technology was invented to solve that one fundamental problem. It's all about finding brilliant new ways to see what nature never really intended for us to see.
Okay, here's our game plan. First, we'll tackle the basics, the absolute physical limits of what we can see with light. Then, we'll get into the clever hacks scientists use to make see-through living cells actually show up. After that, we dive into the world of fluorescence, basically making the things we want to see glow in the dark. We'll meet the rock star protein from a jellyfish that completely changed the game. Then we're going to push past the limits of light itself with electron vision. And finally, we'll see how scientists go from taking pictures to actually taking the cell apart piece by piece. Let's get to it. So let's start with the OG of cell biology tools, the light microscope. For centuries, it was our only window into the cellular world. But as amazing as it is, it runs into some pretty serious hard-coded physical limits. And here's the first key idea you have to get, and it's one people mix up all the time. It's not about magnification, it's about resolution. Think about it like your TV. You can walk right up to the screen, magnifying the image, but all you'll see are blurry pixels. Resolution is what gives you that crisp 4K detail. It's the ability to tell that two things right next to each other are actually two separate things. That's what really matters. And this picture says it all. Look at the left, high resolution. You can see all the intricate little spikes and details on the pollen grain. On the right, low resolution. It's just a blurry blob. That right there is the difference between just seeing a cell and actually being able to understand the machinery inside it. Okay, so this formula gets to the very heart of why we have that limit. This is the Abbe diffraction limit. And what it tells us is that our resolution, our clarity, comes down to two main things. First is lambda, the wavelength of the light you're using, and second is NA, the numerical aperture, which is just a fancy way of saying how good your lens is at gathering up that light. The takeaway? To get better resolution, you need a shorter wavelength and a better lens. But because the wavelength of visible light isn't infinitely small, we eventually hit a hard wall. For light microscopes, that wall is at about 0.2 micrometers. Can't get any clearer than that, no matter how good your microscope is. All right, so we're bumping up against a physical limit on resolution. But honestly, there's a much more basic problem we have to solve first. Living cells are mostly water. They're basically transparent. So how in the world do you see something that's invisible to begin with? If you just stick a living cell under a standard bright field microscope, this is what you get. Yeah, not much, right? It's a ghostly outline with almost no detail. You can't tell what's what. Sure, we could add a stain to see things better, but that usually means we have to kill the cell first, and that kind of defeats the purpose if you want to watch life in action. So how do we get around this? Well, this is where the genius of phase contrast microscopy comes in. It is such a clever solution. See, as light passes through different parts of the cell, some denser than others, it gets slowed down just a tiny bit. That creates an invisible phase shift. What this microscope does is convert those invisible shifts into visible changes in brightness. And suddenly, bam, all the internal structures just pop right out. And we can push this idea even further with something called differential interference contrast, or DIC. This uses another set of optical tricks, but the result is this stunning, almost three-dimensional looking image. You get this incredible sense of depth and texture. And again, this is all done with light manipulation, without a single drop of stain on a living cell. Just brilliant. Okay, seeing the whole cell is great, but what if you're not interested in the whole cell? What if you're hunting for one specific type of protein among thousands of others? It's like trying to find one specific person in a crowded stadium. How do you do it? Well, the answer is, you make them glow. And the magic behind this comes from special molecules called fluorophores. Here's how they work. You hit them with one color of light, say blue, and they get excited. Then, a split second later, they calm down and release that energy by emitting a different color of light, say green.
A fluorescence microscope is designed to hit the sample with that blue excitation light, but then filter it out so the only thing you see is the green emission light coming back. The result is that iconic image, a glowing object on a perfectly black background.
So we can attach these glowing fluorophore tags to things like antibodies to find our protein, but that still often means we have to kill and preserve the cell to get the antibodies in. The real dream, the holy grail, has always been to watch these proteins moving around and doing their jobs inside a living cell. To do that, we needed a true revolution. And that revolution, amazingly, came from a jellyfish. This is green fluorescent protein, GFP. This was so transformative it won the Nobel Prize. What scientists figured out is how to take the gene for this glowing protein and basically stitch it onto the gene for any other protein they're interested in. The cell then does all the work. It reads the instructions and builds your protein with its own little glowing lantern already attached. It was a total game changer, letting us watch the machinery of life happen live. And once you can make proteins glow, you can do some really cool experiments. This one's called FRAP, Fluorescence Recovery After Photobleaching. It's awesome. First, you find a spot full of your glowing GFP-tagged proteins. Then you blast that one little spot with a powerful laser, which basically bleaches the GFP and turns the light off. Then you just sit back and you watch. Over time, new unbleached proteins from elsewhere in the cell wander into that dark spot, and the glow comes back. By timing how long that takes, you can figure out how fast your protein is moving inside the cell.
This is even more clever. We can use GFP to ask, are two different proteins actually touching each other? It's a technique called FRET. Imagine you tag protein A with a blue glowing tag and protein B with a green glowing one. If they're floating far apart and you shine blue light on them, only the blue one will light up. But, and this is the cool part, if they get really close and actually interact, the excited blue protein can pass its energy directly to the green one, like a baton in a relay race. So you shine blue light and you see green light. It's a direct signal that tells you, yep, those two proteins are physically interacting right now.
But no matter how clever we get with light, no matter how many tricks we use, we always slam into that 0.2 micrometer wall, the diffraction limit. To see the really fine details of the cell, the internal structure of organelles, the membranes, individual viruses, we had to give up on light entirely and find something new to see with.
That something new was the electron, and its wavelength, when you speed it up in a microscope, is about 0.004 nanometers. Now let's put that number into perspective because it is staggering. The wavelength of visible light is around 500 nanometers. The electron's wavelength is over 100,000 times shorter. This isn't just a small step up, it's a monumental leap in power. It's why electron microscopes blew the doors off what we thought was possible, revealing an entire hidden world of what we now call cellular ultrastructure. And with this new power came entirely new techniques. One of the wildest is called freeze fracture. You take a cell, you flash freeze it in liquid nitrogen, and then you literally crack it open with a tiny super sharp knife. The cool thing is the fracture plane loves to split right down the middle of the cell membrane, peeling the two layers apart. You then coat that fractured surface with a super thin layer of metal, and what you get is this unprecedented view of all the proteins studded inside the membrane. There's just no other way to see this. Okay, so we have these incredible ways to see the cell's architecture, from its overall shape right down to individual proteins. But to really understand how a city works, you can't just look at it from a helicopter. You need to go into the buildings, see what's inside. We need to take the cell apart piece by piece and study what each component does. But how do you sort the parts of something so ridiculously tiny? The workhorse technique here is called differential centrifugation. The first step is to break open a ton of cells, making a kind of cell soup called a homogenate. Then you put that soup in a centrifuge and spin it. At a relatively low speed, the heaviest, densest things, like the nuclei, get packed into a pellet at the bottom. You then take the liquid on top, move it to a new tube, and spin it again, but faster. This time, the next heaviest things, like mitochondria, form a pellet. You just keep repeating this process, spinning faster and faster each time to isolate smaller and smaller components.
But how do you know you actually got a pure sample of mitochondria? You can't exactly look in the test tube and see them. Well, this is where biochemistry comes in. It turns out that every organelle has its own unique set of marker enzymes that act like a biochemical fingerprint. So you can test your sample for the enzyme succinate dehydrogenase. If it's there in high amounts, you can be confident you've got mitochondria. And it's this powerful combination, the pictures from microscopy and the biochemical data from fractionation, that gives us the full story. These tools have taken us on an incredible journey, from seeing blurry outlines to watching single molecules move in real time. The question has evolved from just what's in a cell to how does this incredibly complex machine actually work? And as our tools get even better, we get closer to that answer every single day.